The discovery of this new antibiotic was really an unexpected result of basic research on how HIV proteins contribute to disease and AIDS. And as a result of studying these proteins, we discovered these novel structures that turn out to work very well against bacterial infections, including the complicated lung infections in cystic fibrosis patients.
Cystic fibrosis affects multiple organs, and those problems, particularly the lung, shorten the lives of patients with cystic fibrosis. What happens is that the genetic defect predisposes patients to infections, and those infections in the lung drive the production of mucus that then blocks the airways and makes it difficult to breathe.
Biofilms are sessile communities of bacteria. In cystic fibrosis, the patients for the longest time we knew they developed these terrible bacterial infections, and part of that is they have defective mucociliary clearance, and so any bacteria are going to be a problem. It was recently discovered that these are actually the bacteria growing in a biofilm community, and so when the bacteria grow in these biofilms, especially in association with living tissues, they develop very high levels of antibiotic resistance to the point that they develop an antibiotic resistance that's higher than what we can solubilize. A very exciting promise with these peptides is that we found in our model system that if we take established biofilms and treat them with a synthetic peptide, we actually see it destroys or eliminates the existing bacteria.
A unique feature of these antimicrobial peptides, as we call them, is that they act very rapidly, and that's because what they do is punch holes in the bacterium. It's like a pin bursting a balloon. It's a very rapid action. While the cystic fibrosis patient was our initial target and a high priority target, we also could look at infections associated with burns, with infections associated with indwelling medical devices such as venous catheters. One last application is in the biodefense realm, in terms of a rapid treatment that would be post-exposure to some kind of bioterrorism bacterial aerosol. They can pull out their nebulizers and immediately treat themselves with this peptide to reduce the exposure from, again, a lethal dose to a maybe subclinical dose of whatever the bioterror agent bacteria is.